So, the right kinds of integrants can have effects on the cytoskeleton, causing changes in cell shape, causing cell movement, and causing it to attach to extracellular matrix surfaces. So how does this work? Several things happen. By an indirect pathway, I don't think I have this fully mapped out yet, but the integrants can activate some types of, I think, still fairly poorly characterized adapter proteins, but ultimately we lead to the activation of 3 monomeric G proteins. There are distant relatives of RAS. One is called Rho, R-H-O. Another is called RAC, R-A-C, I don't know what that stands for. Another is called CDC42. CDC means cell division cycle. It's involved in some aspects of cell division. Okay, these are 3 monomeric G proteins that get indirectly activated through the integrants. Now, what do these guys do? All 3 of them, although they form different types of structures, they stimulate the formation and extension of cells. They stimulate cell extensions, and we'll see how in just a minute. Now, when cells make an extension, what's happening is what we call the actin microfilaments, part of the cytoskeleton, grow longer and they push the membrane out, and they organize into different types of structures. For instance, many people have seen an amoeba, or a video of amoeba. You might see structures like this called pseudopods, especially. And if you look at it closely, you see these things stretching out, and then you see what looks like cytoplasm flowing into it. Some cells put out little tiny spiky things called phylopods. As a matter of fact, that's a place of the cells that put out real long phylopods play a role in the gastrulation of a sea urchin. The cells in that future archaneron put these long phylopods out, grab the bottom of the blastocele, and then contract, helping to pull and in-pocket the future archaneron. So this is a long spiky extension. It's called a phylopod. And then other cells, the entire front end forms a thin membrane that kind of ruffles around. It's called a lamellipod. It's a thin membranous layer loaded up with cytoskeleton, but no organelles because it can't fit into that cytoskeletal network. But basically, all 3 of these are different types of cell extensions that different cells use. A lot of crawling cells like fibroblasts and stuff like that use these large sheet-like lamellipods. Things like amoebas and macrophage in the immune system, you see lots of pseudopods. Other cells, including some white blood cells, put out these long spiky phylopods. But the whole thing is these are all caused by cell extensions caused by the elongation of actin microfilaments. And that is caused by these different G proteins. One of them does the pseudopods, one of them handles the phylopods, one of them handles the lamellipods, and I forgot which is which. But how does that work? Okay, well, indirectly, they activate, it leads to the activation. It's about 2 other steps beforehand, but we don't want to go in. They activate something called the ARP. It stands for actin-related protein, the ARP2/3 complex, which consists of a protein called ARP2, another called ARP3, and several others. Now, what does this thing do? Well, this is a kind of neat thing, and let's put this over here for space purposes. This stimulates microfilament, the formation of new microfilaments on pre-existing ones. and lets those filaments grow real long, kind of like Pinocchio's nose when he lies. Okay, well, microfilaments do that too. So what happens here? Here we have a microfilament. It's made of the protein actin. It's essentially like the thin filaments in muscle. Okay, pre-existing. Now, at one end of it, we have proteins that normally block growth from this end. You can only add new actin molecules, the subunit molecules, the individual proteins, at the ends of the filaments. You can never do that from the sides. So, we have something that's often called a capping protein. And that keeps this filament from growing. Well, various kinds of cell signaling cause the capping protein to fall off, and now new actin molecules add on. And that causes the filament, the microfilament, to grow in length like Pinocchio's nose. Now, one of two ends, the filament, of course, has two ends. One of the two ends is where most of the growth occurs. So it can occur at the other end. But most of the time, it's going to grow at one of the two ends, and the filament grows longer. The cell membrane is here. As this thing stretches out like Pinocchio's nose, it pushes the cell membrane out. Well, that's one thing that cell signaling can do. It can cause the loss of that blocker, that capping protein, what causes it to fall off. But the second thing is where our friend, the ARP2-3 complex comes in. Now, it happens to attach the side of a microfilament like a barrel. And the tip of the barrel is especially good at attracting a small core of actin molecules that allow the rapid formation of a new microfilament. So we have sort of this core we sometimes call the nucleus of an actin filament, and now more actin molecules come together and we have a new microfilament growing. And eventually you have this cytoskeleton, you start getting a kind of network-like structure, continuously branching like so, and all these ends are getting longer and longer. It starts pushing the cell membrane out, and lo and behold, you have just generated a new cell extension. So, this is why cells change their shape, and this also leads to cell crawling. We'll have to see that on Tuesday. One more thing, and we haven't got to that yet because that's a slightly different aspect, is we're also going to lead to the formation of attachment points between the cell and the extracellular matrix. Think of something like a spot weld or a piece of Velcro, a little patch of Velcro that cells have. We call those attachment points to a surface focal adhesions or focal contacts. That's where our friend FAK came in, because it was first found in focal adhesions. It's also found in other places too. But clearly, what we're seeing now, we've seen that stimulating the right integrins with the right extracellular matrix activates the RAS-MAPK pathway. It influences a couple other signaling pathways as well, but especially the RAS-MAPK pathway. That stimulates cell growth, cell differentiation, things like that. Acts like a growth factor. Second, we've seen that this can stimulate changes in cell shape by causing cell extensions through these G proteins, Rho, RAC, and CDC42, through activating that ARP2-3 complex and knocking capping proteins off of microfilaments. So now we get growth here and side filaments coming off the side at an angle. So, cell extensions. What we will see when we come back is going to be how we get these focal adhesions and then put it all together and see how a cell crawls. If you want to see that in advance, look at the YouTube video about me crawling a cell. I want to say doing something to the desk, whatever. But demonstrating cell crawling, and that will give you a heads up. That's funny too. So take a look at that, folks, and I will see you people on Tuesday.